Welcome to the Department of Nutritional Sciences 31st Anniversary Hans Fisher Lectureship. I am Josh Miller, Professor and Chair of the Department of Nutritional Sciences, and it is so good to see you, so many of you in person, as well as those on Zoom. 2 years ago, at the start of COVID, we had to cancel the Fisher Lectureship, and last year it was held virtually over Zoom, and is therefore a pleasure to return to some sense of normalcy and welcome you all. The Fisher Lectureship is one of the highlights of the academic calendar for the Department of Nutritional Sciences and the School of Environmental and Biological Sciences. The lectureship honors the legacy of the founding chair of the Department of Nutrition in 1966, which evolved into the Department of Nutritional Sciences in late 1980s. That founding chair, Dr. Hans Fisher. Dr. Fisher was hired as an assistant professor of nutritional biochemistry at Rutgers in 1954, and his work encompassed tryptophan and niacin metabolism in chicks, assessing the essential amino acid requirements for egg production and the growth of rabbits, a reevaluation of amino acid requirements in adult humans. His work also encompassed histamine, carnosine metabolism, and their roles in wound healing and stress management, as well as the influence of tryptophan and vitamin B6 on serotonin in the brain and associated behavioral consequences, including alcohol addiction and withdrawal. Dr. Fisher's work also informed our understanding of lipid metabolism in relation to atherosclerosis and blood cholesterol in a chicken model. Showing you one of the papers here on avian atherosclerosis and the retardation by pectin by Dr. Fisher and his colleagues. All right, this work led to the recognition that solid fibers like pectin had strong cholesterol-lowering properties that were beneficial in lowering the incidence of coronary plaque formation. It is upon this legacy and his many contributions to the Department of Nutritional Sciences and Rutgers University that we hold the Hans Fisher Lectureship. And it is my honor to let you know that Dr. Fisher is attending today via Zoom from his home in Boston. And please join me in acknowledging Dr. Fisher. Before moving on to introducing our distinguished guest speaker, first let me acknowledge Christina Duffy, whose tireless efforts make the event happen each year, and let me also acknowledge the efforts of our department's seminar committee, including its previous chair, Tracy Anthony, and its current chair, Harini Sampath, as well as the other members, Malcolm Watford, Sue Shapsis, and Joe Dixon. Now, I'd like to introduce to you Tracy Anthony, Professor of Nutritional Sciences, who will introduce our honored speaker.
Thank you, Josh. It is my great privilege to introduce to you Dr. Teresa Davis, who is a professor of pediatrics at the Children's Nutrition Research Center, Baylor College of Medicine. Dr. Davis is internationally recognized for her studies on the nutritional regulation of protein metabolism, as well as the nutrient sensing and metabolic signaling pathways that regulate skeletal muscle growth. Her work has been impactful for many, including me. Her 1986 paper in Biochemical Journal, entitled Response of Muscle Protein Turnover to Insulin After Acute Exercise and Training, explored the impact of insulin on protein synthesis, and what she identified in that paper was a reduction in protein synthesis after exercise. That was a seminal finding of its time, and it laid the groundwork for efforts by my lab and others to look to improve muscle recovery after exercise through nutrition. So Dr. Davis conducted that work with Dr. Irene Carl at Washington University School of Medicine, where she worked as a postdoc and then a research associate and assistant professor. And if you're interested, before then, she actually completed her undergraduate and her graduate degrees at the University of Tennessee. Now, she eventually left Washington University School of Medicine for Baylor College of Medicine. and since that time has risen through the ranks, loyally serving the Children's Nutrition Research Center there. She currently serves as an associate director of the CNRC Postdoctoral Fellowship Program. She also holds adjunct faculty roles at Texas A&M University. Dr. Davis's research program is supported by the National Institutes of Health and the U.S. Department of Agriculture. She has received many awards. I'll highlight a few. So among them are the Stokestad Award from the American Society for Nutrition, the Animal Growth and Development Award from the American Society of Animal Sciences, the Morrison Award from the American Society of Animal Science, the University of Tennessee Centennial Leader Award, and the Baylor College of Medicine Department of Pediatrics Research Mentor Award. She is also an ASAS Fellow and an ASN Fellow. Dr. Davis has served as president of the American Society for Nutrition, as a member of the board of directors for the American Society of Animal Science, a scientific advisor for the International Life Sciences Institute North America Food, Nutrition, and Safety Program, and a member of the 2020 USDA HHS Dietary Guidelines for Americans Advisory Committee. Dr. Davis is currently the editor-in-chief of the Journal of Nutrition and president-elect of the American Society for Animal Science. It is difficult to fully describe how much I admire the life of Dr. Davis, her thoughtful intellect, her grace, and her strength of character. She is an exemplar of interdisciplinary nutrition research and an inspiration to women scientists everywhere. So I know you are looking forward to her talk as much as I am. And so please join me in giving Dr. Teresa Davis our warmest welcome.
So can everyone hear me? Is it coming through the microphone or do I have to talk really? Everybody can hear? Okay, great.
It's not through the microphone.
Oh, it's not through the microphone. So I can just move this up. Outside voice, or the voice I use with my children, right? Whichever one it is. So thank you very much, Tracy. Thank you for that lovely introduction. That was the most beautiful introduction I've ever had. So thank you so much. And it's really, truly an honor to be asked to give the Hans Fisher Lectureship lecture for this year. Hans Fisher is truly a giant in the field of nutrition, not only for his work on amino acid requirements and amino acid metabolism, but also for other work in dietary fiber and cholesterol metabolism, and diet and alcoholic fatty liver disease, and many other areas. So I'm really, truly honored to be asked to give this lectureship and to join many distinguished scientists who have lectured previously for this lectureship. And I'd also like to thank Josh for inviting me, and Tracy and Malcolm and all the other people who were involved in this lectureship. And also, it's really a great pleasure to see so many old friends here in the audience, that I've known for, over 20 years and so forth. So I'm really greatly honored to be here and to talk to you about some of the work that I've done along with a lot of my collaborators on the nutritional regulation of growth in neonates. So let's see. So the work in my lab has focused on the regulation of growth during the neonatal period. So we focus on growth during the neonatal period. Can I bring up a pointer or is that just the mouse? Okay, that's fine. So my work has focused on the regulation of growth during the neonatal period because growth during this period of life is greater than in any other stage of postnatal development. So my lab is really focused on the regulation of growth in skeletal muscle, because skeletal muscle is the most rapidly growing protein compartment in the body during early life. And because of this rapid muscle hypertrophy, skeletal muscle is a primary determinant also of amino acid requirements in the neonate. So growth is really rapid during this early stage of life. On the other hand, there are many infants that are born in this country and really across the world that are born of a low birth weight. And they're born prematurely. So premature is being defined as being born less than 37 weeks of gestation, as opposed to 40 weeks, which is generally considered to be term. And in fact, there are 15 million infants that are born preterm every year across the world. And when you look at that map, you can see that preterm birth is particularly prevalent in Africa, Southeast Asia, but it's also quite prevalent in the U.S. as well. And so here's a map of the United States where you see that 1 in 10 infants are born prematurely. And this is particularly prominent in the southern part of the United States, probably because healthcare tends to be not as good in that part of the U.S. So is preterm birth a problem? Well, yes, it is. It's a leading cause of child death. In fact, premature birth accounts for almost 20% of all deaths in children less than 5 years of age. And recent data during the COVID pandemic suggests that if a mother is diagnosed with having COVID during her pregnancy, that she's more likely to give birth to an infant prematurely. Now, although the survival has increased in these preterm infants in recent years due to neonatal technologies and drug treatments, morbidity still remains quite high. And these preterm infants are likely to have feeding difficulties. They have difficulties in sucking, swallowing, and breathing. And they're also more likely to have vision, hearing problems, and developmental delays. And so most of these preterm infants are discharged from the hospital weighing less than the 10th percentile for intrauterine growth standards. So they're more likely to be growth restricted even after they're born. And some of them are even small into adulthood. And these preterm infants are at increased risk for many long-term developmental outcomes, which are problems such as cognitive deficits, motor impairments, reduced work capacity. But really important for the discussion that we're going to have today is they're likely to have an altered body composition. And so they're likely to have a lower lean body mass. Fat mass may be the same or increased, but it's the lean body mass that's reduced. So that creates a high fat to lean body mass ratio, and that probably contributes to their increased risk for obesity later in life, insulin resistance, type 2 diabetes, and cardiovascular disease. Now, because ethical consideration limit our ability to conduct mechanistic studies in human infants, mothers don't like us taking a sample of their baby's muscle, we have been using the neonatal pig as an animal model. And it really is a great animal model because the anatomy of the baby infant, the human infant, and the baby pig is very similar. The anatomy is similar, the metabolism is similar, and the body size is large enough that you can use a lot of different experimental approaches that you could not use in that tiny little mouse pup. So you can repeatedly sample blood. You can do tracer kinetics across the hind limb or the portal drain viscera as an indication of muscle metabolism and gut metabolism. And of course, you can alter the feeding modality. So you can feed them parenterally, that is by IV, or you can feed them by orogastric tube. You can vary the diet in all kinds of ways. And you can't readily do that in newborn mouse pup. So using that neonatal model, we determined now quite a few years ago that feeding stimulates protein synthesis in neonatal pigs. And that response decreases with development. So this is just a figure that shows protein synthesis rates. This is in skeletal muscle, so I'm going to be talking about muscle today. And this is over the first month of life. And what you see is in these newborn pigs that are just fasted for a few hours and then fasted and then given a meal, the protein synthesis markedly increases. But as they get slightly older, you can see that response to feeding decreases with age. And so what's the mechanism by which feeding stimulates protein synthesis? So now, quite a few years ago, we developed this technique, which we call the pancreatic substrate clamp technique, which we can not only control the glucose and insulin, but we can also control the amino acids. Because, you know, if you infuse insulin, amino acids are going to dive as well. And amino acids are necessary for protein synthesis. So using this technique, what we did was to infuse insulin, to raise insulin from the fasting to the fed level, while we kept amino acids and glucose at the fasting level. And we found that that insulin could stimulate protein synthesis. Infusion of amino acids alone increased protein synthesis. But if we combine the two, raising insulin and amino acids together, there was no further increase in protein synthesis. But importantly, that increase in protein synthesis with insulin or with amino acids was much greater in the newborn pig than in a slightly older animal, even though those two ages of animals saw exactly the same amount of insulin and exactly the same amount of amino acids in the blood. They responded differently. And also importantly, the insulin and amino acids increased protein synthesis up to the fed level. So this showed us that ingestion of food increases protein synthesis in muscle. And it does so by raising the insulin and amino acids in the blood, and that insulin and amino acids then signal down to protein synthesis in muscle, and that response decreases with development. So how does the neonate then translate those nutrient signals and those hormonal signals into anabolic signals to stimulate growth? So first, what I'm going to be doing is just showing you an overview of the insulin and amino acid signaling pathway. And then we're going to talk about individual components later in the signaling pathway. So just kind of give an overview of it. So studies in our lab, put together with studies that have been conducted by a number of other people, including Dr. Anthony, have shown that ingestion of food causes a rise in insulin in the blood. That insulin then signals down through the insulin signaling pathway to activate mTOR-C1, or mechanistic target of rapamycin complex 1. When mTOR-C1 becomes activated, it then activates the translation initiation signaling pathway that regulates the binding of the messenger RNA to the ribosome, and together with the factors that regulate the binding of methionine tRNA to the ribosome, increases synthesis of protein. So studies in our lab have shown that the abundance of many of these positive regulators of the insulin signaling pathway and the translation initiation signaling pathway are much more abundant in muscle of the newborn than in muscle of the slightly older animal. But perhaps even more interesting is that rise in insulin in response to a meal profoundly activates these signaling proteins, and that response decreases with development. Now we know that feeding also causes a rise in amino acids in the blood, and that amino acids then also signal down to mTOR. And there have been a number of models that have been proposed, and this is the most accepted here. But what I want to illustrate is that the abundance of many of these signaling proteins is greater in muscle of the newborn, and that decreases with development. So let me just give you some examples of this profound developmental change in the activation of these signaling proteins. So we'll just give an example. First, looking at the insulin signaling pathway, where ingestion of food causes a rise in insulin in the blood. This insulin then binds to the insulin receptor, activating the receptor, which then associates with IRS1, or IRS1 associates with the insulin receptor, which then activates PI3 kinase, which brings AKT to the plasma membrane, where it becomes activated. And then AKT activation then causes the phosphorylation and deactivation of the inhibitor TSC2, which then releases its inhibition of mTOR. So looking at first at AKT phosphorylation as an indication of insulin signaling, we can see if we raise insulin just from the fasting up to the fed level. So we're interested in what happens in response to feeding. This is not illness, chronic illness. This is not, nutrient deprivation. We're just interested in what happens in response to feeding. So sort of a normal situation here. So when we raise insulin from the fasting up to the fed level, we can see that AKT phosphorylation profoundly increases, but that response decreases as the animals get a little bit older. And when we move down the signaling pathway to TSC2, we also see a phosphorylation of TSC2, which decreases with development. And as you would expect, there's no effect of amino acids. Now let's move down the signaling pathway to mTOR-C1. So mTOR can be activated by insulin, but also by amino acids as well. So when mTOR becomes activated, it increases the phosphorylation of S6K1 and 4E-BP1, which increases the binding of EIF4E with EIF4G, which binds to the messenger RNA, increases translation initiation, and increases protein synthesis. Okay, so what did we find when we looked at the translation initiation signaling pathway? We found that raising amino acids from the fasting to the fed level increased mTOR phosphorylation. Raising insulin alone increased mTOR phosphorylation, and that was then transmitted down the signaling pathway to S6K1, to 4E-BP1, the association of EIF4E with EIF4G, and that decreased with development. Even though those two ages of animals saw the same amount of insulin, the same level of amino acids, they responded differently. So I think that data and others showed that the feeding-induced stimulation of protein synthesis in muscle is due to the activation of both the insulin signaling pathway and the amino acid signaling pathway. That leads to activation of mTOR and stimulation of translation initiation, but that response is developmentally regulated. So how rapid is that change in the rate of protein synthesis after a meal? Because all the data that I showed you was taken at just one point in time, usually about an hour after a meal or an hour after a clamp. So Fiona Wilson, who was a postdoctoral fellow in the lab, who came to us from the University of Aberdeen, looked at the insulin levels in the blood in response to a meal. So this meal provided 1/6 of the daily nutrient requirements. She looked at what happened over 1/6 of a day, which is 4 hours. And you see that insulin rapidly rose, peaked about 1 hour, but then fell rather rapidly, well before 4 hours. Amino acids also rose rapidly. Their elevation was more sustained, but they fell down to about baseline by 4 hours. But what we were particularly interested in was the translation initiation signals and how they change in response to feeding. And so now looking at the insulin signaling pathway, as indicated by AKT phosphorylation, we saw a rapid increase in AKT phosphorylation after consumption of the meal, but then it rapidly falls down to baseline well before 4 hours. Okay, and then when we went downstream to mTOR phosphorylation, which is getting input from insulin, but it's also getting input from amino acids, you see a rapid increase, just like with AKT, but it's more sustained. But it does fall down to baseline by 4 hours. And so as a result, we see a similar trend with the protein synthesis, a rapid increase that's sustained for at least 2 hours, but then falls down to baseline. Okay, well, that's interesting, right? But why do we really care? Why do we care about the time course of the change in protein synthesis after a meal? We care because I got really concerned when I saw that data, because there's a lot of preterm infants that are not meal fed. They're fed continuously at a constant flow rate. That insulin and amino acids are not spiking. So I was concerned because I said, well, if insulin and amino acids are not spiking, are the signaling pathway factors not being activated, and is protein synthesis not going to be going up to the same degree that you would after a meal? So we wanted to determine whether this meal feeding promoted protein synthesis and led to an increase in growth. So first, let's take a look at how premature infants are fed. Well, premature infants generally are fed by parental nutrition initially, at least to some degree. And then they're promoted then to enteral feeding as soon as they can tolerate the enteral feeding. And when they're enterally fed by nasogastric or orogastric tube, they're fed either continuously or they're fed by intermittent bolus feeding, which is like meal feeding. So it's not really known which way is better. Is continuous feeding or intermittent bolus feeding better? You know, which mode of feeding is better, and particularly, which mode of feeding is better at promoting growth? So Carolina Gazzaneo, who was a postdoctoral fellow in the lab, she was from critical care medicine, wanted to take on this question. So what she did was to take baby pigs and she fed them first continuously for a 24-hour period, and found that, as you would predict, insulin levels were very low and constant. And then when she took another group of pigs and she fed them by intermittent bolus feeding, like meal feeding, what she found was that insulin levels, of course, spiked after that meal was given and then fell down. And then when you give another meal, they rise and then it falls down. And so you see this cyclical pattern of insulin, which you might predict. And the same is the case for amino acids as well. But what happened with the signaling proteins that regulate protein synthesis? Well, first, let's take AKT as a representative of the insulin signaling pathway. And what you see is if they are continuously fed for a 24-hour period, AKT phosphorylation is a little higher than in the fasting condition, although you can see it's not statistically significantly different. Then she looked at the bolus-fed pigs. So first looking at what happens after a 24-hour period. Now, this is before the meal is given. You know, so it's given every 4 hours. So at this 24-hour mark, it's just before a meal is given. And you see AKT phosphorylation is low. She gives them a meal, and then, as you would expect, AKT phosphorylation increased markedly. Then she went downstream looking at some of the translation initiation factors, here represented by the association of EIF4E with EIF4G. And you can see again this marked increase that occurs after a meal is given. So our primary outcome in this experiment was protein synthesis. What happened with that? So we were quite happy that we saw that with continuous feeding for a 24-hour period that protein synthesis is increased above fasting. And that's comforting because they are getting nutrients. We know that babies that are continuously fed, they do grow, so protein synthesis is increased. Now, if they're bolus fed and we measure protein synthesis right before the meal is given, you see protein synthesis is greater than in the fasting condition, but it's the same as what it is with continuous feeding. But if you give them a meal, protein synthesis increases. So that's very good and exciting. But what we ultimately want to know, right, is does that promote growth? Because that's really our ultimate objective. So Samer El-Kadi, who is a postdoctoral fellow in the lab, wanted to take on this question. So he fed baby pigs either continuously or by intermittent bolus, which is like meal feeding, for 21 days. And what he found is that if they were bolus fed, that they had a higher body weight. They grew more, so they had a higher body weight gain. And he could detect this even by, I think, 9 days of age. Even though those animals were fed the same amount of diet. So they were more efficient at utilizing their diet for protein deposition. It's just amazing. And then he also found that the linear growth was greater in these bolus-fed pigs. Not only was body weight increased, but lean mass was also increased by this meal feeding pattern. And fat mass, they increased in their fat mass, but the increase in fat mass was similar in the continuous and bolus fed. So this increase in body weight was due to an increase in lean gain. And this increase in lean gain, as you would predict, was due to an increase in the weight of different muscles. And these are muscles of different fiber types. So it's not just in the fast twitch glycolytic, but it's also in the slow twitch oxidative muscles. So I think those data really clearly showed that if these baby pigs are bolus fed, that this induces this pulsatile pattern of insulin and amino acids that activates the insulin and amino acid signaling pathway that stimulates protein synthesis and then enhances lean growth. But if they're continuously fed, those insulin and amino acids are low and constant. And that mutes the insulin and amino acid signaling pathway, it blunts protein synthesis, and restricts growth. So you would say, well, then all premature babies should be fed intermittent bolus fed, right? I mean, why feed them continuously? Well, some premature infants have to be fed continuously because they're meal feeding intolerant. You can't bolus feed them. And so we wanted to develop some strategy to improve the growth of these infants that have to be continuously fed. So to do that, we turned our attention to our favorite amino acid, leucine. And so based on the work that we had done in our lab and that Dr. Anthony had done in her lab, we tried leucine as a strategy. So we know, of course, that leucine is an essential amino acid. It can act as a signaling molecule to stimulate the intracellular signaling pathways that regulate protein synthesis. And studies in our lab and others had shown that leucine is that primary driver of that increase in protein synthesis after a meal. So here's just an example of one of the studies that we conducted. And so this, Jeffrey Escobar, who is a postdoctoral fellow in the lab, infused leucine just to raise leucine just from the fasting to the fed level, not pharmacological levels, but just an increase up to what you would typically see in the fully fed state. And that leucine alone stimulated protein synthesis. But if he gave an equimolar amount of isoleucine and valine, no effect. And you can see why. Leucine stimulated the association of EIF4E with EIF4G. Isoleucine and valine had no effect. So what we postulated is that pulsatile delivery of a leucine supplement during continuous feeding could enhance lean growth by basically mimicking that pulse in leucine that occurs after a meal. So Claire Boutry, who is a postdoctoral fellow in the lab, who is now at Nestle in Switzerland, took on this question. And so she took baby pigs and she fed them continuously for 21 days. And every 4 hours she pulsed them with with leucine. With leucine. And of course we need an isonitrogenous control, so she pulsed the other group with alanine. So, same amount of energy, same amount of protein, same amount of nitrogen. And so what she found was that these baby pigs that were pulsed with leucine increased their body weight more than those that were pulsed just with alanine, despite the fact that they consumed the same amount of diet per kilo body weight. And this increase in body weight was due to an increase in lean gain. And interesting here, fat gain was lower in these leucine pulsed pigs. And so, isn't that what you want to do to promote lean without promoting fat? And, another thing that we gather from this is there looks like there's a repartitioning of nutrients toward lean and away from fat. This increase in lean gain, as you would predict, was due to an increase in muscle weight, which was due to an increase in protein synthesis, which was due to an activation of the translation signaling pathway. So that data and others suggested to us that leucine can enhance lean growth by stimulating mTOR-C1-dependent translation initiation signaling pathway. That leads to protein synthesis in muscle of neonates. So we think that leucine supplementation can be a potentially useful adjunct to the nutritional management of neonates in order to promote their growth. Now, all of the studies that I just described to you were conducted in baby pigs, but they were born at term. So what we're really interested in is how can we promote the growth of low birth weight and preterm infants. So shouldn't these studies be conducted in the preterm pig? Yeah. So a few years ago, we turned and developed a preterm piglet model along with Doug Burren to test whether prematurity alters these protein metabolic responses to nutrition. So, what should we hypothesize of what prematurity does? I spent a lot of time talking about this profound response to feeding and insulin and amino acids in the newborn, which decreases with development in pigs that are born at term. So should we hypothesize that if these baby pigs or human infants are born prematurely, should they have a more profound, a more robust response, just continuing on that developmental track? Because we do know data in fetal rats, that fetal rats, they're not born yet, so the fetal rats have a higher rate of protein synthesis than newborn rats. So we could hypothesize they have a higher response. Or we could hypothesize that they have a lower response because there's data in premature infants that when they're measured at term equivalent age that they have a lower lean mass. So maybe they have a reduced response. So we hypothesize that the protein metabolic response to feeding is lower in the preterm than in the term. So Jane Eberheis, who is a postdoctoral fellow in the lab, wanted to take on this question, and what she did was to take pregnant sows and deliver the baby, or deliver the preterm pigs at a time that's equivalent to 30 weeks of gestation in the human infant. So she delivered some by C-section prematurely, and then some others right at term. And then she gave them total parenteral nutrition for 3 days. And then after that 3-day period, she fasted them for just 4 hours, and then she gave them a meal. So we had preterm and term pigs that were fasted, or they were postprandial, that is, they were fed. So here you see body weight in the preterm and the term pigs, which you would expect. The preterms are going to be smaller, right, than the terms. But what's important is that the relative weight gain, that is, the gain in weight per kilo body weight per day, was lower in the preterm than in the term, despite equivalent nutrient delivery. And then after 3 days, muscle weight, at least for some of the muscles, was lower in the preterm than in the term, but some of the organs were higher in weight expressed per kilo body weight. And then we first looked at the insulin levels in the blood. And of course, if they're fasted, it's very low and constant. But then if these these term pigs are given a meal. As you would expect, there's a rapid rise in insulin. And the term pigs are in the orange. In the blue are the preterms. So you see a slower rise in insulin in the blood of these preterm pigs, although they come to about the same level. Then when we looked at the glucose and the different amino acids here represented by leucine, you also see a slower rise in the amino acids and glucose levels in the blood, although they came to about the same level. So what about protein synthesis? In this experiment, that was our primary outcome variable. And so what you see here is protein synthesis in response to the meal in the preterm and in the term. And you can see that there is a response to feeding in the preterm and the term, but it's lower in the preterm and the term. And that occurs in muscles of different fiber types. Okay, so what are the key regulatory molecules involved in this differential response of muscle to feeding in the preterm versus the term? Okay, so first we're going to look at some of the components in the insulin signaling pathway, particularly AKT phosphorylation and TSC2 phosphorylation. And so what we can see is that there is an increase in the phosphorylation of AKT on these two sites, which are critical for activation of AKT, but the response is lower in the preterm than in the term. And that feeds downstream to the phosphorylation of TSC2. So let's look at amino acid signaling. So I haven't talked quite as much about that. And so there's different models that have been proposed for amino acid signaling. This is the most accepted, such that when amino acids are transported into the cell, they then activate mTOR via these RAG proteins, which bring mTOR to the lysosomal surface where they associate with regulator proteins as well as Reb being actually activated by the insulin signaling pathway. So sensors have been identified for some of the amino acids, in particular leucine. And cestrin-2 has been identified as a sensor for leucine, such that leucine can bind to cestrin-2, and that pulls cestrin-2 away from GATOR-2, so that GATOR-2 can then bind to GATOR-1 and cause GATOR-1 to release its inhibition of mTOR. So mTOR becomes activated. So we looked at some of these signaling components, and we couldn't pick up any effect of prematurity on this association of the cestrin with GATOR, but we did for looking at the RAG proteins and the association with mTOR, as well as the association of Reb with mTOR. And then looking at mTOR signaling, both mTOR phosphorylation, the phosphorylation of S6K1 and ribosomal protein S6, as well as the phosphorylation of 4-EBP1, we can see a reduced response in the preterm versus the term. So I think we can tell from this that prematurity blunts that feeding-induced activation of the insulin and amino acid signaling pathway that regulates protein synthesis in muscle. And that probably contributes to the extrauterine growth restriction that's seen in many premature infants. But I think many of you are probably thinking, okay, Teresa, you just showed me data that showed that the rise in insulin, the rise in amino acids in the blood was a lot slower in these premature pigs than in the term pigs. So we had to say, well, you know, is this reduced response in the preterm, is it due to a decrease, in secretion, a decrease in the insulin, lower insulin that the muscle is seeing, or is it true resistance at the end organ level, at the level of the muscle? So in order to test that, Marco Rudar, who is a postdoctoral fellow in the lab, took this question on and delivered pigs by C-section, and then performed the clamps that we talked about earlier, such that we controlled insulin levels in the blood, amino acid levels in the blood, and then compared that to the fasting condition. So here you can see we achieved our targeted glucose, insulin, and amino acid levels in these pigs, and they were the same in the preterm than in the term. And here we see that insulin increased the phosphorylation of components in the insulin signaling pathway, but that was lower in the preterm than in the term. And that was the case also for the amino acid signaling pathway, looking at the association of the RAG proteins with mTOR, a lower response in the preterm and the term. And that feds downstream to looking at the translation initiation signaling pathway, a lower response for both insulin and amino acid in the preterm than in the term. And that led to lower rates of protein synthesis in the preterm than in the term. So I think those studies clearly demonstrated that prematurity does cause resistance of the insulin signaling pathway, the amino acid signaling pathways that lead to mTORC1 activation and protein synthesis, and that probably contributes to the reduced lean growth in prematurity. So I've mentioned a lot of postdoctoral fellows that have enabled this work to happen. And so I'd like to also mention Marta Furato and Doug Burren, as well as Agus Siriawan and Han Nguyen in my lab, and also Scott Kimball, that I've collaborated with a number of times over the years, as well as I've been really fortunate to have support by NIH and USDA. And so I live here, literally live here, in the Children's Nutrition Research Center at Baylor College of Medicine, so that it is located in the Texas Medical Center, which is the largest medical center in the world. You know, there are a lot of medical schools and medical facilities that are there. This is the building that we're in, so that's about right here. And I think this is an old picture, so it's grown up quite a bit since then. This Children's Nutrition Research Center also houses the editorial office of the Journal of Nutrition. So the Journal of Nutrition was created in 1928. So we're almost 100 years old. And I was not editor back then. But I have been editor for about 8 years. And so this is the cover of the first issue of Journal of Nutrition way back in 1928. And here's the cover, I think this is the March issue that I was laying on my desk, and I snapped a picture. I love this picture. It's been around for a long time. In fact, the American Society for Nutrition was created in order to house this journal. So it's truly the journal that launched a society. The Journal of Nutrition is the oldest nutrition journal of the world. We publish original research manuscripts focusing on nutritional mechanisms, but we also publish reviews and editorials. And we really try to promote the science that's published in these journals and highlight various papers that are published in the journal by publishing summaries. On the ASN landing page, we have a dedicated writer that tweets every day the articles that are published in Journal of Nutrition. We have a very strong impact factor. We're on the top ranked science journals. We are fast. So we're as fast as some of the predatory journals. So less than 20 days to first decision. So we really are fast. But I can't leave you without saying beware of the predatory journals. So I have a lot of young people here in the audience. I know, some of the longer, more established people know this, but really you got to beware of these predatory journals because we're getting emails like every single day. It seems asking us to submit your paper, to this journal. We guarantee you're publishing. We need some more papers here to publish in the next month. So please submit your work to that journal. that you've never heard of, nobody else has ever heard of. And they're soliciting people every day, to be on the editorial boards, to be a guest editor, and so you were getting, all these spam emails to solicit manuscripts from these predatory journals. But these predatory journals frequently have fake editorial boards. I gave a lecture at one of our other prominent universities right before the pandemic. And then one of the professors that I know really well came up and said, "Look, Teresa, look, I Googled my name, and look here. I'm on this journal I never heard of. I'm on the editorial board." And so is my wife. So that happens quite a bit with some of these predatory journals. And sometimes they get so many publications that they become now kind of in the established journal world. Even they started out as predatory journals. And, they'll say they're going to peer review your paper, but it's not peer reviewed. And you really, peer review really does strengthen your work. So you've got to really watch out for these, because they publish with titles that are so close to our established journals that you can't tell the difference. Even old established authors or some, like us are scratching our heads saying, now, is this JBC or is this something else, So we don't know. And so these publishers are really exploiting the open access model. This is publishing for profit, to put money into the pocket. This is not publishing with your society journals. This is putting money into the pocket of a publisher. When you publish in society journals, you are promoting the scientific community, because any money that those scientific societies make from those journals, they're putting it back into you. They're putting it back into meetings. They're putting it back on giving scholarships and fellowships to young scientists. They are promoting your work. These other journals are making money, for their stockholders. So, how do you avoid these predatory journals? Well, don't answer their emails, for one thing. I think I get an email from one of the journals, I think about every day, wanting me to be guest editor, editorial board, whatever. And so it used to be that we could go to this list of Jeffrey Beall. And so on this list, he had almost 100 publishers, almost 100 standalone journals, but that was taken down in 2017. So he had on that list publishers like Omics that was sued by the Federal Trade Commission, and the Federal Trade Commission won. And so he had some of the other publishers, like the publishers that the publisher of Nutrients was on this list as well. And so how do you know that you're submitting your work to a trusted journal? Well, do your colleagues know the journal? Do you know the editorial board? Do you know the publisher? Go to a journal that you can trust. And you can trust, who can you trust? You can trust your society journals, whether it be the American Society for Nutrition, the APS, or pediatric research journals, or some of those. I would encourage you, go to your society journals, because you are strengthening your society. So I would please submit your work to Journal of Nutrition. I invite you to submit your work there. And these are some of the covers last year. So any questions, I'm happy to take them.
Thank you so much for coming to lecture to us. And we have this commemorative plaque. It reads, "In recognition the Hans Fisher Lectureship, Nutritional Regulation of Muscle Growth in Neonates, Teresa A. Davis, Ph.D., April 6, 2022. Thank you very much.
Thank you so much,
Appreciate it. And
Thank you.
round of applause.